So let me do a recap. Okay. So remember, in photosynthesis, what is very important is, you having a knowledge about what are light harvesting pigments. Okay. So plants on their leaves have what we call light harvesting pigments, as I put here. Okay. So light harvesting pigments are also known as light trapping pigments. Okay. So each pigment, okay, one pigment can only trap light within a certain range of lambda, certain wavelength. So within that range. So this is actually a limitation to the pigment, because sunlight comes in a very wide range of wavelengths. So sunlight comes in a wide range of wavelengths, somewhere around 430 nanometers to about 700 nanometers, which ranges from violet light up to the red light, which I've already put in sequence and order. So it has that. But one single pigment, all right, will not be able, one single harvesting pigment, a light harvesting pigment, cannot harvest the entire range, cannot, because it can only harness certain range of wavelength only. So that is a limitation on the one pigment. So it cannot harness all wavelength of sunlight. So how then your leaves are adapted? Because if the leaves only happen to have one pigment, that limits the leaf from trapping the sunlight and carrying out photosynthesis at an adequate rate. So instead of having one pigment, the leaves are adapted to have many pigments. So usually leaves have two or more pigments. All right. So by that way, they are able to harness the entire range of wavelength of sunlight. For example, leaves could have chlorophyll pigment, which itself comes in the form of A and B. Like chlorophyll A harnesses violet region, light from the violet region and white from the red region. Whereas your B harnesses more towards the blue region and more towards the orange region. So by having A and B, this range and that range is covered, leaving behind only green and yellow. That is where your carotenoids and phycocyanin comes in. The other pigment comes in to harness this part of your wavelength, this range. So by having this pool, let's say by having these four pigments, a single leaf is able to harness the entire range of wavelength of light available or given by our sunlight. All right, that is why this understanding is important. So in the nutshell, plants have two or more light harvesting pigment to ensure that they are able to harness the full range of wavelength of sunlight and to carry out photosynthesis at a very high rate. Okay, now let's shift our focus now to two spectrums that are important, which are still related to what we call the light harvesting pigment. All right, so there are two spectrums that you need to know. Okay, related to this light harvesting pigment. What are they? They are what we call the absorption spectrum and the action spectrum. So now we are going to look into that. So we have two types of spectrum based on that. So we have what we call the absorption spectrum. And we have what we call the action spectrum. These are again related to your light harvesting pigment. And of course, all these are related to your photosynthesis. So first, we got to know the difference between the absorption spectrum and your action spectrum. That's the first thing. Then the second thing is for us to know how these two spectrums correlate. That means the connection between the two. We got to know that. So remember what I told you. The focus is we have to know the differences between the two. After which we got to find the correlation between the two to see what information or data can we extract from both these two spectrums or spectra which are joined together. All right, the combination of the two. That is what we are going to look at. All right, let us look at the absorption spectrum. So absorption spectrum is a spectrum, all right, which basically refers to a graph that is obtained when we plot light intensity against wavelength. Okay, light intensity against wavelength. So this light intensity refers to the light intensity absorbed by the leaves. Okay, absorbed by the leaves. Okay, light intensity absorbed by the leaves. versus your wavelength. Okay, versus your wavelength. So you will find that we have just a simplified diagram. Okay, this will refer to chlorophyll A. Okay, or this refers to your chlorophyll, and then somewhere here you have something like that. You have your carotenoids coming in like that. Your carotenoids are coming like that. In fact, I'll remove this part. Okay, your carotenoids should be more like that. Okay, then other people will come like that. Okay, these are all the light intensity that's absorbed. So that will be more of hypocyanin, and that's carotenoid. So this one, referring to carotenoids. This is chlorophyll. That means I've combined your A and B together. Chlorophyll. I've combined your A and B together. So let me put it in a different color so it's easier for us to see that. It's just a sketch. Okay? We don't want an exact diagram, just a sketch. But of course, the sketch also is okay. This would be phycocyanin. All right. Okay. This is your absorption spectrum. So that means your leaves, the intensity of the light that is absorbed by leaves in this range is done by chlorophyll. So that absorption shows that chlorophyll absorbs light intensity, absorbs very high light intensity. That means chlorophyll is able to absorb within this range, and the amount of light it absorbs is quite high. And then carotenoids are able to absorb light within that range. So that's the highest peak or the intensity that it is able to absorb, and that goes for phycocyanin. Now, what's your action spectrum? Your action spectrum is a graph that is obtained by plotting rate of photosynthesis. By plotting rate of photosynthesis with lambda. Okay? So now you can see the difference between the two. Action spectrum is rate of photosynthesis with lambda. Okay, rate of photosynthesis is lambda, whereas your absorption spectrum is light intensity absorbed by the leaves. So if you were to look at the rate of photosynthesis absorbed by sunlight, you see, first we'll look at what it is first. Then you'll find the correlationship between the two. So if you were to see, you will find that it would be something like this. Okay, it is more like that. So this is your action spectrum. All right. So what does this action spectrum tell you? That means a high rate of photosynthesis occurs within this range of lambda. Within this range of lambda, this wavelength is suitable for photosynthesis. That means that means lambda within this range is suitable for photosynthesis. Okay, why? Because it promotes a high rate of photosynthesis. You can see for yourself. That means the photosynthesis rate is one of the highest there. So definitely this lambda must promote photosynthesis. And the second range of lambda that is important for photosynthesis will be this. This is second most suitable lambda for photosynthesis. Suitable lambda for photosynthesis. So that is what this spectrum tells you. It tells you which lambda is suitable for photosynthesis. So lambda within this range, lambda within this range. Okay, so in terms of light, this is in the violet blue range. This is more towards the orange red range. Okay, so therefore light which are violet to blue and orange to red are the most suitable light for photosynthesis. Okay, not that others are not suitable. You still have photosynthesis in other range, but these are the most suitable. And now we correlate to that. Now, which of the pigments here, the three pigments here, absorbs light within this lambda and within this lambda? Which of the pigment over there absorb light within this lambda and within this lambda? So your answer is very clear. Your answer is quite clear. It is, that's the lambda we're looking for. That's the lambda we're looking for. And my question is, which pigment absorbs light within that lambda, which corresponds to your photosynthetic, what you call rate, which is high, the lambda that is required for photosynthesis. So it is, of course, chlorophyll. So from there, we make the conclusion, among the three light pigments, chlorophyll is the best pigment to carry out photosynthesis. That is why we always talk about chlorophyll when associated with photosynthesis. Okay, so these two spectrum combined gives us that information that chlorophyll is the most suitable pigment as a light harvesting pigment to carry out photosynthesis. But it's not the only pigment, but the most suitable pigment. So that is supported by the action spectrum and the absorption spectrum. All right, so I hope you understand the correlation between the two spectra or the two graphs that you have in front of you. So tell me now. So the conclusion I'm going to make from the correlation. Conclusion from correlation. Will be okay.